Hello and welcome to lecture 15 and 16 regarding cell nucleus and chromatin structure. These lectures were given by Professor Gabor Sabo. So, what is chromatin? Chromatin is DNA plus histones plus non-histone proteins in the nucleus, which are together these three called chromatin. And chromatin, as we said, contains the hereditary information, which is DNA. We have two main types of chromatin, heterochromatin and euchromatin. And what are the differences between heterochromatin and euchromatin, which is very important? Heterochromatin is a condensed form of chromatin. So chromatin itself is condensed. It's a very condensed form of DNA. But heterochromatin is even more condensed. It's very condensed. Whereas euchromatin is the condensed form of chromatin, comparing to heterochromatin. And euchromatin is transcriptionally active, whereas heterochromatin is transcriptionally inactive, which means that when genes are getting on and off, during transcription, when genes are getting transcribed, and then out of these genes we will have mRNAs after transcription, and these mRNAs will go to the cytoplasm and then will be translated to proteins. These things happen in case of euchromatin genes, because these are euchromatin genes that can be transcribed. That means that they are transcriptionally active, whereas heterochromatins cannot be transcribed, which means they are transcriptionally inactive. And the reason is because of the condensation. So the more condensed a chromatin structure is, the less probable will it be for that chromatin to be transcribed. And I really want you to understand that concept, because if you understand it, you gonna know the very concept of gene regulation. So when we're talking about gene regulation, when we will talk about gene regulation, you will know that when we have a more condensed form of chromatin, a more condensed form of DNA, all these transcription factors and these proteins which are needed for transcription cannot bind to this very condensed chromatin, because it's a mess and they cannot find that sequence which is specific to them. And that is why they cannot, these transcription factors and RNA polymerases and other proteins which are necessary for the transcription, they cannot find those sequences on heterochromatin or condensed form of chromatin, and the transcription will simply not take place. So one other thing about heterochromatin that you need to know is that heterochromatin itself divides into two categories: constitutive and facultative heterochromatin. Well, constitutive heterochromatin is kind of giving what it is in its name, that it is always a heterochromatin. So constitutive, no matter what, this type of heterochromatin remains heterochromatin. This type of heterochromatin will remain always transcriptionally inactive. And where we can find these constitutive heterochromatins? On the telomeres or around the centromere of chromosomes. Now, if you do not know where the telomeres or centromeres are, I'm going to show it to you further in this lecture. So what is the difference between constitutive and facultative heterochromatin? Well, facultative heterochromatin is a type of heterochromatin that under specific situations, either like genetically situations or epigenetic situations coming from outside the gene information recorded in the genes. So under a specific situation, these facultative heterochromatins are able to become euchromatin. So they are able to decondense. And why do we need that? Why do we need facultative heterochromatin? Because let's say there's a cell in your brain and there are specific DNA sequences that a cell in the brain needs to transcribe, whereas a cell in your liver doesn't need to transcribe those specific set of DNA sequences. So what happens is that those set of sequences in your brain cell will be euchromatin, will be active, whereas the same sequence of DNAs will be inactive in your liver cells. Because as you know, we have in each and every of our cells, we have all the genes. We have all of our genes in all of our cells. But the thing which makes these cells different is that what parts of those genes are getting transcribed and what parts of those genes are on and what parts are off. And facultative heterochromatin is an important aspect of that. And also you probably know that the fluid which fills the nucleus is called nucleoplasm, the same way as the one which fills the cell itself is called cytoplasm. So we said that chromatin, no matter if it's hetero or eu, is condensed to a certain extent. And why is that? Why do we need? Because that was a question in one of our exams. Why? What is the main reason? What is the most important reason that DNA is condensed? And the reason for DNA being condensed is that if it's not condensed, it will not fit in the cell. So that is the most important reason. Other choices that you may have in the exam are also true. But the most important reason is because if DNA wasn't condensed in form of chromatin in our nucleus, it wouldn't fit in the nucleus because it's a very long molecule, DNA. So, and there are different levels of condensation for DNA. Well, the first level is DNA itself, and its diameter is 2 nanometers. These diameters are important. These are one of the few exceptions in cell biology that numbers are important. That the DNA double helix itself is 2 nanometers. Then we'll have histone proteins forming these octamers, forming this 8 subunit structure around which DNA will wound. And these structures, like the DNA with the histone proteins together, are called nucleosomes, and that will cause further condensation of DNA. And the average size of these structures, which are histones plus DNA, is 11 nanometers. And these 11 nanometer structures, which in this slide it's not shown their diameter, which is 11 nanometer, they are called solenoids. So 11 nanometer solenoid. That's also important. It was in an exam. And then these the nucleosomes will condense more to a 30 nanometer structure, and condensation will continue. There will be other proteins like condensins, which I think is easy to remember. Its name is condensin, and protein and the matrix scaffold proteins, which help the further condensation of DNA to a 300 nanometer structure. And as you can see, when the DNA reaches the 13 nanometer structure, now we have got chromatin loops. So chromatin loops, we have them, the first time that we can see them is in 300 nanometer structure. the condensation continues furthermore to 700 nanometer, which is chromatin itself. And even chromatin can be condensed more, even heterochromatin can be condensed more to chromosome. And why do we need this overcondensation? That is because we want to divide the cell, and we're going to talk about it in cell division in details. Let's talk more about nucleosomes. As we said, a nucleosome is a structure which is formed by an octamer histone core, octamer because there are 8 histone proteins in the core, which a DNA strand, the DNA molecule or DNA double strand wounds around this structure 2 times. And the length of the DNA going around each nucleosome is about 200 base pairs. That is also another important number, 200 base pairs around each of these histone octamers. And the DNA which is linking each nucleosome together, these DNAs are called linker DNA. And as you can see, there's also a yellow structure here. This yellow structure, this yellow thing is also a protein. It's also a histone, but it is histone H1. And histone H1, we cannot find it in the core of nucleosome, but we can find it usually on the linker or around the linker DNA, sometimes on the nucleosome itself. But one thing for sure, it is not in the nucleosome core. And the name of other histones which form the core are histone H2A, H2B, H3, and H4. 2 of each of these. So we have got 4 different histones, different types of histones in the core, and out of each of them we have got 2. And histone H1 is not in the core, but it is on the nucleosome or in the linker DNA. Now it's a good time to talk about topoisomerase enzymes. You probably by now know the basics of transcription or replication because you had genetics and you studied them basically in details in genetics. You don't need to know them in that detail in cell biology. But one thing you probably know is that when RNA polymerase is acting during transcription, the 2 strands of DNA, they need to be separated for a temporary period of time, and that will allow the RNA polymerase to do its job. But one problem that can occur when you simply open up the DNA strands is that along the DNA molecule there can be overwinding, there can be something of a messy structure can occur along the molecule, because you can imagine it as there are like 2 strings in perfect helical form, and you just pull one of them or 2 of them or both of them away at a point, and you can see at other points there will be like knots occurring. And that the same thing can happen in our DNA molecules. And the enzymes which are responsible for compensating this this phenomenon, for not letting this phenomenon to destroy the structure of DNA, the enzymes are topoisomerase enzymes. What these topoisomerase enzymes do is that they break and then rejoin these DNA molecules or these DNA strands further along the DNA molecule, well, the RNA polymerase is acting. So when RNA polymerase is acting, they break the DNA strand so that this overwinding doesn't occur. And when the action of RNA polymerase in a certain part is over, they then rejoin or re-ligate that specific site that they break. But the thing that you really need to know for the exam, the important part of topoisomerases that you need to know, is that topoisomerase 1 cleaves and religates single strand of DNAs. So it breaks and rejoins single strand DNAs. Whereas topoisomerase 2 cleaves and religates or breaks and rejoins double strands of DNA. So these two, that the topoisomerase 1 is for single strand, topoisomerase 2 is for double strand, are the important features of topoisomerases that you need to know for your exam. So now I'm going to talk about something that most of you know either from genetics or high school or even secondary school. You've always been told that like we've got two different type of chromatin. There's a chromatin that we've got in interphase, and there's a chromatin, a form of chromatin that we have got in metaphase. And the chromatin that we have got in metaphase is no more called chromatin. It's called chromosome. It's a very condensed form of chromatin, and there are other differences. Like for example, the chromatin in interphase consists of one gigantic linear DNA molecule and proteins which are attached to it, but one DNA molecule. Whereas the chromatin or chromosome in metaphase has got two DNA molecules. And the reason is because during cell cycle, in the S phase of interphase, chromatin or DNA replicated itself. So when we're talking about metaphase, we've got two DNA molecules and they are super condensed. As a matter of fact, the chromosome in metaphase is the most compact form of DNA. And now you can probably guess that it is definitely that this type of DNA, when we're talking about chromosome, is definitely inactive, transcriptionally inactive, because as we said, the more condensed a DNA structure is, the less transcriptionally active it will be. So if even heterochromatin is transcriptionally inactive, then chromosomes are definitely transcriptionally inactive. And one other thing about the chromosome structure, as I said, about those of you who don't know where the telomeres or centromere is, if this is our chromosome here, you can't see centromere here. So in the middle, well, the two chromatids are attached together. So each of these replicated the DNA molecules are now called chromatid, and together they form a chromosome. And these two sister chromatids, because they are for one chromosome, are attached at the point called centromere. And the ends, and they have, of course, arms and legs. And at the end of their arms and legs, their ends are called telomere. So telomere, centromere. And the areas around centromere and at the telomere are composed of constitutive heterochromatin. There is one more thing about this lecture, and that is we talked about condensins. They help chromosome condensation to form, well, those DNA loops which we talked about, those chromatin loops. And there is cohesin. So we said these sister chromatids are attached at one point, at centromere. And how is that they're just attached at that point? Is that because of cohesin. Cohesin is a specific type of protein which acts like a glue. And since you probably know that during a cell cycle we need to separate these sister chromatins, we need to get rid of cohesin somehow, and how we're going to talk about that in cell cycle lectures. So we talked about the structure of chromatin. Now that you know how the chromatin is the place where genes are, you are getting introduced to how these genes can be regulated. Again, you're gonna study them in detail in genetics, but here we're gonna emphasize on those parts important for cell biology. One of the ways to regulate transcription of a gene is via position effect variegation. And what is position effect variegation? It is when a gene which is usually active, which means it is usually in euchromatin form, is translocated near a heterochromatin. And because of this translocation and getting close to a heterochromatin, other elements of transcription, like transcription factors, like DNA polymerases, they consider this gene as part of that heterochromatin, and so this gene will be off. So again, to put it in a more formidable way, is that position effect variegation is when gene expression is arrested because a gene which is usually active and in euchromatin form is translocated near to a heterochromatin structure due to the invasive spread of heterochromatin itself. Because, well, the heterochromatin is getting bigger and it is now kind of occupying the euchromatin genes. A good example of that is the white gene of Drosophila. So Drosophila is a fly, I think, or something like that. And you can see here is the eye of that Drosophila. When it is usually active, it is completely in this orangish color. When the white gene is completely active, the eye color is completely orange. When, due to position effect variegation, white gene is inactive, we will have these white spots on the eye of Drosophila. Now, one other thing which really has got a very important role in regulation of gene expression, that what part of DNA should be transcribed and what part of it should not, which basically is telling genes on and off, is how much of a decondensed structure that that DNA has at the moment. So the accessibility of genes are very important for transcription. And in order to understand that the thing which I just said better, the lecture slides they introduce a new thing, DNA's the sensitivity. What is DNA sensitivity? It is the relatively high sensitivity of transcriptionally active regions of chromatin against certain nucleases. So when we say transcriptionally active, we mean the condensed part, the part which doesn't contain that much of a nucleosome around them, because as we said, nucleosomes help for condensation. So those parts which don't have that much nucleosome, those parts of DNA, are more sensitive against nucleases, against enzymes which basically hydrolyze DNA. So the more active a site is, the transcriptionally active a DNA site is, the more it will have sensitivity against these nucleases. And an important example of one of these nucleases is DNase 1. And you can see in a place where it is on the nucleosome itself, here, like here, we've got this flash, which means that this flash, which means that this DNA, due to its very high condensation, is DNase resistant, is very resistant to DNase enzymes. And then we've got like these linker DNAs which are not on the nucleosome but very close. So these are DNase sensitivity sites. And we've got these parts which are not protected at all. They're completely like naked DNA. And these are DNase 1 hypersensitivity sites. And the more a site is sensitive to DNase, for example, these hypersensitive sites, the more transcriptionally active that would be. And so going back to our main point, that is a way for regulating gene expression. You don't want one part to be active, you condense it. You want a part of DNA to be active, to be on, genes to be on, you decondense the structures there. So we said the more decondense a chromatin structure, the more active. And condensation and decondensation has got a lot to do with nucleosomes. So the places with more, the places on DNA with more nucleosomes, those sites are more condensed. The sites with less nucleosomes are less condensed. And how are these nucleosomes spread along DNA? Just randomly, or are there specific sites, or are there sites with that are always with no nucleosomes? Well, these can be all the case. We've got random spread of nucleosomes, that there are sequences that are not involved in regulation. In those sequences, we've got random distribution of nucleosomes. But when we're talking about inactive genes that have to be inactive at that moment, nucleosomes are fixed there. There are specific sites for them. And there are, of course, sites with no nucleosomes. That at sites with active genes, we don't need nucleosomes there. And these are interchangeable. So a site which is active can become inactive. So nucleosomes can form there. So if in an exam you saw a true or false question that the distribution of nucleosomes along the chromatin is always random, is always fixed, these are false. It depends on the activity of the gene, and it depends on a lot of other things. But one of the main things that controls this distribution of nucleosomes, that if, for example, in a certain tissue, specific types of cell, they want to turn on a specific sequence of DNA, how does the cell get rid of nucleosomes on that site in order to make that site accessible? Well, that is via chromatin remodeling. Chromatin remodeling is an ATP dependent modification. So if it is ATP dependent, its action, which is chromatin remodeling, is energy dependent. It requires energy. So again, chromatin remodeling is an ATP dependent modification of chromatin structure during turning genes on and off. And there's a very good picture in our slides. You can see on the right side, which is the important part, you can see we've got these nucleosomes on our DNA, on our chromatin, which really make these sites inaccessible for transcription factors, which we're going to talk about. These transcription factors basically help the oversimplified version is that they are good for transcription, for turning genes active. But we've got all these nucleosomes here, they can't be transcribed. So we've got this enzyme, chromosome remodeling enzyme, which comes and binds on those sequences and uses energy, uses ATP hydrolysis in order to get rid of these nucleosomes on those sequences and make those sequences accessible and active. Well, another factor which can really influence the gene regulation is epigenetics or epigenome. And epigenetic is a sum of factors influencing gene activity that are over the information stored in DNA. So when we're talking about epigenetics, we don't have direct changes in the information stored in the DNA. So we don't have, like, for example, when we're talking about a change in base sequence or that kind of stuff, that is, we're talking about genetics. But when we're talking about epigenetics, we're talking about a level above that. So basically it's a landscape of histone and DNA modifications, mainly of these two. So instead of changing them innately, we're gonna modify them by, for example, phosphorylating them or acetylating them or methylating them. And a very important subdivision of epigenetics is histone code. Histone code is basically referred to different modification, post-translational modifications of histones, like acetylation, methylation, and phosphorylation. These reactions happening to histones, first of all, they are post-translational modifications, and they are from the subcategory of epigenetics. And the very important thing is that some people may think that because it is over the information, because epigenome or epigenetics is over the information stored in DNA, it can't be hereditary. Whereas the truth is, it can be hereditary. So epigenetic is hereditary, and that is a very important detail they like to ask about it. One thing that you need to know for cell biology, of course, because you need to know all of these in more detail in genetics if you're taking the subject, is that whenever we're talking about methylation, that is bad for transcription. So methylation usually always inhibit the transcription, usually always inactivate the transcription. Whether it wants to be methylation of histone or methylation of DNA, and the enzymes which perform methylation are called methyltransferases. The same way, enzymes performing acetylation are called acetyltransferases, and these enzymes work the opposite. Acetyltransferases and acetylation works the opposite of methylation. So it helps transcription. So acetylation helps activation of a gene sequence. It helps turning a gene on. And phosphorylation isn't really that important. What you need to know about phosphorylation is that phosphorylation doesn't always, usually, actually, the truth is that phosphorylation, as far as I know, in most of cases doesn't fully inactive or active transcription of genes. So if they gave you as a true or false question in the exam that phosphorylation inactivates the transcription, they probably mean that the main function of phosphorylation is inactivation, and that is wrong. That is definitely wrong. The process which its main job is to inactivate is methylation. One more thing that I need to talk about, although again most of you probably already know, is the action of transcription factors, enhancers, and silencers, and promoters. So first let's start talking about promoters. Promoter is a sequence of DNA located 5 prime upstream from the genes. So it is located before the genes which it is regulating. So it is located before them. 5 prime upstream means before a certain thing, comparing to other like sequences on the DNA. And it's responsible for a starting of gene transcription. So the important thing about promoter that you need to memorize: 5 prime upstream from the genes, and the site which transcription starts. For starting of transcription, what do we need? The thing you probably know, all of you, is RNA polymerase. So RNA polymerase needs to bind to these promoters to start the transcription. But the problem is RNA polymerases usually cannot single-handedly find these promoters. They can't, because DNA is very big and they just floating around, don't know where exactly to bind to. And that is where the action of enhancers and transcription factors come to play. Now, enhancers, in contrast, enhancers or silencers, which we're going to talk about silencers just in a moment, in contrast to promoter, they can be located either upstream, downstream, and they don't need to be just away from the genes. They can be 100 base pairs or 1,000 base pairs away from the genes that they regulate. But there is one the condition that they need to have, is that these enhancers or silencers, they need to be on the same chromosome with the genes that they want to regulate. And the way they act, enhancers, is that a specific transcription factors bind to these enhancers, and when they are activated, a loop will form in DNA, as you can see here, to simply transport these transcription factors and enhancers together to the site of transcription. And what the transcription factor enhancer complex does is that it facilitates the recruitment of RNA polymerase. So now those RNA polymerases, which we said it's hard for them to find the promoter, now they can find the promoter with the help of enhancers and transcription factors. So the only thing which remains is the silencers. Now, what are silencers? Exactly like enhancers, they are DNA sequences. They can be 100 or 1,000 base pairs away, exactly like enhancers, but their function is the opposite. So they help. So when certain transcription factors bind to those silencers, they help to inactivate gene transcription. And that is the main difference. And one thing that I really need you to know is that the people in the department really like to ask about is that transcription factors are proteins, whereas enhancers and silencers and promoter are gene sequences, are DNA. Their monomers are DNA. And if you know that, you're good for this lecture. And this was the end of this lecture. See you in the other one.